This is Dr. Dave Septel, Director of Research and Development for the Macular Degeneration Foundation and editor of Macular News. We're thrilled to have Professor Joel Schuman here from the University of Pittsburgh. Professor Schuman, could you tell us a little bit about your current position?
Yes, thanks for having me. I am the professor and chairman of ophthalmology at the University of Pittsburgh, director of UPMC Eye Center, and I'm also director of the Louis J. Fox Center for Vision Restoration.
Tell us about that center.
The Fox Center is dedicated to the discovery and translation of new methods of applying regenerative medicine to ophthalmology. And by that I mean taking work from the laboratory all the way through to actual clinical implementation. Our goal is to improve the vision and lives of patients who have visual loss. And so we want to be able to reverse that disease and not just treat it. We have a number of different types of approaches to this, going from stem cell approaches to small molecules to gene therapy, as well as technology solutions for dealing with vision loss or blindness. We have investigators who are working on stem cell therapy for the retina, for retinal regeneration for a number of different diseases, but very importantly for macular degeneration. We have scientists who are working on optic nerve regeneration. Also scientists who are working on regeneration of the cornea, and in the cornea you have several different portions that do different things. So there's the surface of the corneal epithelium. There is the kind of the inside of the corneal sandwich, the stroma, and there is the back of the cornea, which keeps the cornea clear, and that's called the corneal endothelium. The corneal epithelium is actually being treated with stem cell therapy now, live and in patients. The corneal stroma is where you generally see corneal scars that compromise vision, and there is a group of scientists at UPMC Eye Center at the University of Pittsburgh who discovered a stem cell that is for the corneal stroma, and they actually are in the process of translating that discovery to clinical patient care with collaborators in India. And then the corneal endothelium has been a real challenge. It does not grow in culture, but through collaborations with engineers at Carnegie Mellon University, we've been able to grow corneal endothelial cells in culture by changing the actual firmness of the substrate on which those cells are growing. Finally, in terms of stem cell approaches, we have a couple of scientists who are working on stem cell therapy for glaucoma, and specifically for the trabecular meshwork. And the concept there is to regenerate the trabecular meshwork and restore normal function, and thereby cure the high pressure that is associated with glaucoma. In terms of other technology approaches, have an approach using a device called the brain port, and the brain port is it's a totally non-invasive device that has a video camera that goes to a computer, which goes to a small paddle that the individual wears on their tongue, and so they can put the paddle in their mouth or take it out. When the paddle is sitting on the tongue, it provides an electrical stimulation in the pattern of the image captured by the video camera. So somebody who's totally blind, let's say that they're a soldier and they've actually lost their eyes, someone who has no vision at all could experience the visual environment through this sensory substitution approach. It's very exciting, and I'll have to tell you, when I first heard about it, I thought it was bunk. But it's very real. It is the real thing, and people are finding that they're able to kind of reconnect with the world because now they can experience the visual environment, whereas they could not before. And it gives them independence and ends that shut-in phase that is so often a consequence of severe vision loss.
I think it's remarkable that you're addressing this particular unmet need. Can you speak a little bit about the way in which you coordinate activities across these different approaches?
So the FACS Center really provides an infrastructure for collaboration within the world of regenerative ophthalmology. Now, that's a world that didn't exist a few years ago, and we're very excited to be some of the pioneers in creating this type of environment across all aspects of regenerative ophthalmology. What we've done is to build on the model that was created by the McGowan Institute for Regenerative Medicine. And the McGowan Institute has been very successful in developing ways of either regenerating tissue or regenerating function that are useful in other parts of the body and not in the eye. We saw that there was a need in ophthalmology and that we had this expertise right on campus at the University of Pittsburgh. And the McGowan Institute also is based on creating an environment for collaboration. And so we have been able to leverage the skill sets, the expertise of experts from the McGowan Institute and apply them to work in the eye. One of the things that we've done is to create something that we call an Otero Fellowship, Ocular Tissue Engineering and Regenerative Ophthalmology. Through these Otero Fellowships, we have somebody with expertise in ophthalmology and somebody with expertise in regenerative medicine working together to train a postdoctoral student. And that postdoctoral fellow then, we hope, will go on to actually be an expert in regenerative ophthalmology. So kind of growing the future right at home. And we've been successful with the Otero Fellowship so far, and we have several people who have gone on to actually pursue careers in regenerative ophthalmology. And we're excited that this program seems to be working.
I think that's a critical component, training the next generation of people to take the baton and run towards the finish line. In terms of your overall approach, this is clearly a key component. Can you talk about some of the translational efforts that you're undertaking?
Sure. And I have to point out that translation of work, especially biologicals, is very difficult, and you need to make sure that they're safe and effective, and safety first. And the US FDA does impose some very strict rules with regard to the testing of biologicals in patients. And so we have to adhere to those rules. One of the things that we've done is to work with people who are already up and running in working with biologicals either in the US or elsewhere. And so I mentioned that one of our scientists, James Funderburg, who is working on corneal stem cells, has partnered with a group in India at LV Prasad in Hyderabad. And they've been able to now get to the point where they're ready to use these stem cells in humans. So this is a finding that went from a discovery in a laboratory at University of Pittsburgh right through to the point now where that discovery is about to be put into humans. An interesting thing about this is the excitement that a basic scientist actually gets when their discovery in the laboratory is actually going to go into clinical use. Because many scientists work their whole lives and don't see the application of their discoveries. And so this is great not only for the patients, but for the people who are doing the basic kind of creative discovery research work, because it gives them really a perspective on the impact of their research. It's sometimes easy to become disconnected and to feel that you're just doing research to do research, but we're doing research to actually help patients.
I think that's an enormously important and valuable contribution, making sure that scientists are motivated and can see the results or the progress in the clinic for the work that they're doing in the laboratory. Professor Schuman, thank you so much for being with us. If there's anything else you'd like to add at this point, please do so.
I very much appreciate the opportunity. I do want to mention that the FACS Center is an incredibly important part of what we do in the Department of Ophthalmology. The Department of Ophthalmology, though, does have a large range of types of research, and there's research going all the way from basic immunology of the eye to ocular imaging. And many of these kind of more traditional modes of research also have application within the regenerative ophthalmology sphere. And so there's a lot of interaction even within the department, collaborations between investigators who are using different techniques or investigating different things in order to actually push forward the regenerative ophthalmology program as well as the more traditional ones.
On the issue of immunology, there's been a lot of discussion about the use of embryonic stem cells and whether the eye is truly a sequestered site that is immune from the effects of the immune system, so to speak. What are your views with regard to this?
And my understanding is that the embryonic stem cells actually are immune suppressive. Surprisingly, they tend to survive and integrate in environments that might otherwise be hostile to foreign cells. And Jim Funderburg, in his work in the cornea, found that human stem cells, when injected into mice with cloudy corneas, actually did not cause an immune response, but rather went in, set up shop, rebuilt the cornea so it was clear, and were still present after a long period of time, and did not seem to stimulate any inflammatory or immune reaction.
I think that's a very critical component. Thank you again, Dr. Schuman, for an extraordinary interview and a terrific overview of the great work that you're doing.
Thank you very much.